All right, so we are going to start this first section of notes. So in terms of where we are and where we're going, this first unit is not very long. It's just basically an introduction to sort of science and how science works. We'll also talk a little bit about some of the tools we use. We'll also talk about the statistics we use frequently in science. And this will help us as we're moving through the rest of the year as we come back and talk about planning experiments and things like that. So you can think of science. So the unit's called scientific inquiry. Inquiry just means sort of like asking questions. So when we think about science and what science is, it's sort of two things. You could think of it as like just a body of knowledge. Like this textbook here is a genetics textbook from college, and it has a whole bunch of information in it. Scientists have learned over the last 100 years about genetics. So sometimes people think that that's what science is, a bunch of facts about the natural world. And that is part of what science is. These are all scientific hypotheses and theories that have been tested over time. But science is also something else. It's a way of learning about the world. It's a way of asking questions and answering those questions. It can help us learn new things. You guys just didn't plan an experiment, carried it out, and you learned something, and you learned a little bit about what the slugs from my backyard, what foods they prefer. So you conducted some scientific inquiry. Now back in 7th grade, you learned about problem, research, hypothesis. So that's kind of a simplified way to talk about how science works. Sometimes in the earlier grades, we talk about the scientific method. Scientists do this step, then they do this step, then they do this step. It's really very artificial. Scientists can follow lots of different ways of learning new information, and they can learn new things in a variety of ways, not just asking a question, coming up with a hypothesis, doing an experiment. So it's much more varied than sort of we sometimes explain it. One type of science is called discovery science, where basically scientists are kind of collecting large amounts of data and then looking for the patterns in that data. That doesn't really follow the same hypothesis, experiment, observation, conclusion setup, but that's still science. It's still one way in which we can find valuable information. The sort of more traditional way we talked about it is hypothesis testing. That's when we talked about problem research, experiment, observation, conclusion. Those steps are more about testing hypotheses. So that's still important, and that's what we did in this experiment. But we started by asking a question about what slugs prefer. You guys made your predictions. You tested the predictions using an experiment. You looked at your results and saw how they compare to what you predicted. Made a conclusion about whether or not your hypothesis was correct. And then if you continued on, okay, you could have made another, if your hypothesis was incorrect, you could have made another one, tested that, or you could have asked a new question. So these are sort of the steps that we follow today. These are steps that many scientists use as they're trying to learn new things. So let's talk about some of those. So scientific inquiry is how we find new knowledge. We often start by asking a question. Now, science doesn't just answer any question. I mean, you're not going to ask a question and then, what is the meaning of life? Which movie is better, The Godfather or Goodfellas? Those aren't questions you answer
The Godfather.
in science, right? Those are opinions or philosophical questions. We answer questions, we ask questions in science that we can answer through experimentation. So here are some examples. We're going to follow these examples through the next few slides. So we might say, how does the amount of nitrogen fertilizer affect the height of pea plants? That's a very clear question. We could easily set up an experiment that would help us to answer that question. Does increasing the temperature of an enzyme increase its rate of reaction? Do high doses of vitamin C prevent people from getting the common cold? All questions we could ask and answer using experiments. From there, we often will have to generate our hypothesis. And a hypothesis is really our prediction. Something we predict and can test in an experiment. We often will write a hypothesis as an if-then statement. If I change this, then this will happen. Hypothesis has to be a statement. It can never be a question. For example, on the Regents exam, a common question they ask is, what would a possible hypothesis be from this experiment? And in the grading notes, they always say, do not allow credit for hypothesis in the form of a question. Your hypothesis has to be a statement, a prediction. So if I'm thinking about those same experiments that I just asked, those questions I just asked, here are some hypotheses. If pea plants are given additional nitrogen, then they will grow taller. If the temperature of salivary amylase is increased to 40 degrees Celsius, then the enzyme activity will decrease. Individuals who are given 200% of the RDA vitamin C will be less likely to contract the cold. Those are all hypotheses that we can test. And we use an experiment to test the hypothesis. It's just basically a procedure that you use to test a hypothesis. So we use controlled experiments to test our hypotheses. And in a controlled experiment, you always have at least two different groups. Call them the control group and the experiment. What's the purpose of a control group? Why do we need that? Control group.
To show the difference between the two.
Yeah, if we just had a single group and we were changing something, we wouldn't know, well, what would the effects have been if we didn't change it? That control group is the group we use for a comparison. We just keep it under sort of typical normal conditions. We don't change the independent variable in the control group. For example, in our pea plant experiment, the pea plants that we give would get only water and no extra nitrogen. That would be a good control group. The experimental group is the group in which we do change the independent variable. We alter it in some way. Sometimes we might have just one experimental group. Other times we might have 3 or 4 or multiple. We might change the independent variable at different degrees. And once we have our experimental and control groups, it allows us to compare them. So the variables. You guys remember which variables which? No?
Yeah.
Some, yes, no. Did you have to use these in earth science? Did they ask questions about these?
No.
Yeah. No, okay. So independent variable, dependent variable, variables held constant. So the independent variable is the variable that you change, the factor that you alter. A good experiment has just one independent variable. We only change one thing. In our examples, it could be the amount of nitrogen fertilizer, the temperature of the enzyme, the amount of vitamin C people are consuming. Those were the independent variables for those experiments. The dependent variables, these are the data you collect, the information that you're going to use when you make your conclusion. So it might be the height of the peatlands, the amount of enzyme activity, the number of people that actually got the cold. Those are the examples of the dependent. And then we have the variables we would hold constant. So if we're trying to make everything the same except for one factor, it means there's a lot of things we have to keep track of that we try to make sure are constant between our groups. In that peatland experiment, we'd have to have the same soil and water and the same type of plant. In the enzyme example, we want the solution to have the same pH in both groups. In our vitamin C experiment, we want the patients to be the same age and diet, same health level. Only if we hold as many of those variables constant as possible if we have a well-designed, well-controlled experiment. As our experiment progressing, we make observations. Now sometimes we collect observations as the experiment is going along. Other times we might just collect our data at the end of the experiment. But observations are basically collecting area. We could be measuring things. Could be counting things. Using tools to analyze the results. Sometimes we have quantitative observations, which have a number with them. Other times we have qualitative observations, which do not. Sometimes we show our information in a data table, or in a graph, or a chart. So some examples. How tall the pea plants were. Measuring the height of the pea plants every 3 days, those would be our observations. Measuring how much gas the enzyme is producing. Determining how many people contracted the cold during the experiment. Those are our observations. We use those observations to make our conclusions. Conclusion is when we take our data, we might use some statistics on it to say, was our hypothesis supported or was it not? We don't really say that our data proves our hypothesis. We don't say that our hypothesis was true. The way we phrase it is, the data supports our hypothesis or does not. When we're making our conclusions, we're usually comparing the control group to the experimental group to see the effect. We said we might use statistics to test if it's significant at a certain level. Science, we often use 95% as the level of confidence level. So some examples of conclusions. The pea plants are given fertilizer through an average 8.3 centimeters taller than control plants, the hypothesis would be supported. Or celery amylase at 40 degrees had lower enzyme activity than the control group, hypothesis supported. But sometimes our hypothesis is not supported. Saying there's no difference in the number of people that got the cold, our hypothesis rejected. It still is helpful, it's still useful, still learn information from a hypothesis that has been rejected. If our hypothesis is supported by our data, okay, we might repeat the experiment to confirm the results. We want to make sure our experiment is valid. And often what scientists then do is they publish their research. So scientists write a paper describing their experiment, telling about the methods they used, describing the results, providing the conclusions, and they publish these in what we call journals, where other scientists read these. So if I'm a botanist, okay, I might subscribe to a journal, where every month I get this journal. It has, 10 or 15 research papers by other botanists. And it's how I learn, well, what are other people learning in my field? Or I might review their experiment and see if I find any problems with them. This is called peer review. Other scientists, experts in the field, are reviewing the research and evaluating it. Sometimes our hypothesis is rejected, but again, we still have learned new information. In the peer review process, what scientists are often looking at is this a valid experiment? When we say valid, we're saying, is this a well-designed experiment? Can we trust the results of this experiment? And certain things are important in planning a valid investigation. So we need a large sample size. The larger, the better. So the experiment you guys were doing with our slug had a very low sample size, right? You just had a single slug eating from whatever foods you provided it. You could certainly improve that if we had 10 petri dishes, 10 slugs, and you use the same foods. If you saw the same results in most of those, then you could be a lot more confident in the results. Okay? So the number of samples is important. Gives you greater confidence. We want to make sure we have just one independent variable, that everything's the same except for that one single independent variable. And we often might repeat experiments to confirm that the results hold. So if you found that your slug didn't prefer tomatoes or pasta, and you repeat the experiment, you find the exact same thing again, well, now you can be more sure of your answer, more confident of the results. Okay, we'll finish up with this. Just look at this example experiment. I know it's similar to one we were using. So an experiment's conducted of 30 pea plants. 10 are given no fertilizer, 10 are given 5 grams, 10 given 15 grams. After 14 days, the height of the plants was measured. Go ahead and answer those questions about this experiment. Probably only take you a couple minutes. Please. Stirring up your memory from 7th grade.
All right. What hypothesis is being tested in this experiment? A possible hypothesis is being tested. Doesn't tell you specifically, but you can figure it out. Christopher?
If the plants are given more fertilizer, then they will grow taller.
Yeah, that would make sense. If pea plants are given more fertilizer, then they'll grow taller. Or maybe they thought there wouldn't be a difference, but either one is fine. So in this experiment, what's the independent variable? Leah?
The amount of grams of fertilizer.
Yeah, how much fertilizer they're getting. That's the thing that's different between my groups. What about the dependent variable? Anabar?
The height of pea plants.
The height of the plants. The thing we're measuring, the data we're collecting, is the height of the pea plants. What would be some variables that we have to control? Mateo?
Plant at the time.
The type of plant, the time they're given. Dan, you got another one?
The amount of water.
The amount of water they got, the amount of light. Yeah, there's a whole bunch of things we could probably think of. All those things need to be changed. Which is the control group in this experiment? Joe?
The one without fertilizer.
Yeah, the one that just got water and no fertilizer. That's the control group. What about experimental group, Drew?
One with fertilizer.
Well, there's three actually. So yeah, the other three that were getting fertilizer, they would be considered the experimental groups. You can have more than one experimental group. How can we make this experiment better? David?
Have a group of 10 grams of fertilizer. Make it up to 5.
I don't know if that would improve it that much. I think there's a more obvious improvement. Tab?
Test it again.
We'll test it again. Increase sample size. Rather than have 30 plants, you have 60. That would make it a better experiment. Yeah, those are all possibilities. All right. Any questions about anything? We'll finish up tomorrow. We'll do our statistics because that's what we're going to be working on in class.